The cell membrane lives on the edge of life, and it functions to separate each cell from its surroundings. So basically, we could say each cell is like a country, and we have trillions of it in our bodies. And just like actual countries that we know, they each have their border, and you can't just waltz in, but you have to show some form of identification first. And this is where cell signaling or cell recognition comes in. But first, let me take a sec to recognize you guys, of course, my subscribers and viewers right now. This is Yanu Medic, and you're welcome to yet another awesome video. Before I go on, you might want to know that the cell membrane could also be called the plasma membrane or plasma lemma. So I just thought I'd put that out there. The version of the cell membrane that is accepted today is the fluid mosaic model, and it was developed by two American scientists, Singer and Nicholson, in 1972. And you can picture a cake, and then you stick strawberries in it and handles and basically everything that we stick in cakes. Then you should get a picture of what we're talking about, except its cell isn't flat, it's like a blob, sort of like a raw egg without a shell. And so these things that we stick in the cake are the proteins and cholesterol and glycoproteins and so on. And our cake is the massive expanse of the phospholipid bilayer. I already talked about phospholipids in a previous video to give us some sort of basic knowledge about that, so you can quickly check that out if you haven't yet. Just click that. A typical cell membrane is about 5 to 10 nanometers thick, and it is made up of lipids, proteins, and carbohydrates. They're usually in different proportions depending on the kind of cell it is that we're looking at. Also, there are three major types of membrane lipids, and they are cholesterol, glycolipids, and of course, yours truly, the phospholipids. A quick note though, the functions of cell membrane are usually the cumulative functions of each of its components. Okay, so lipids and proteins are the major portions of any cell membrane, and phospholipids are usually the major types of lipids. Phospholipids are unique in their ability to form a bilayer, and basically this happens when they arrange themselves in such a way that the hydrophobic tails are facing towards each other and the hydrophilic heads face outward into the surroundings or into the cell. Therefore, the hydrophobic tails don't have to interact with water because the heads already did that. So I could say the heads are like bouncers, and the underage kids trying to get into the party are the water molecules, or basically anything that isn't small or non-polar enough to get through the membrane. And then the party, of course, is happening in the hydrophobic region of the tails, and definitely some kids will still manage to sneak in, but just a few. So now we have established a stable and efficient barrier that protects the cell from its environment, but therein lies the problem. Some molecules are lucky enough to pass through this barrier without much hassle because they're usually really small or non-polar or both. But the cell cannot live on these guys alone. It has to interact with its environment. It has to send other things in and out that are not just this tiny non-polar stuff. So how does that happen? And this is where membrane proteins come in, the superheroes of the cell. In a land of life and a time of inaccessibility, the destiny of a great cell rests on the shoulders of a young molecule. Its name? Proteins. Or more specifically, transport proteins. There are two major types of membrane proteins, and they are the integral and the peripheral proteins. Integral proteins are the type that are permanently attached to the cell membrane, like you couldn't remove them without destroying the structure of the cell membrane. But peripheral proteins are only temporarily attached, and their major function is for cell communication within the cell or with other cells. So what it does basically is it attaches to a protein or a lipid or something temporarily. It gets a signal in the form of proteins usually, and then goes off to another protein or lipid or whatever to deliver the signal. I know I'll probably have to make a separate video specifically for membrane proteins, but for now I'll just look at some general characteristics. Transport proteins here are the integral proteins, and they have two major types, channel and carrier proteins. And the major difference between these two types is that carrier proteins change their shapes slightly during transport, while channel proteins do not. Oh, and by the way, transport proteins are usually very specific about what they transport. So much so that the glucose transporter will not transport fructose, and fructose is just a structural isomer of glucose. And aside their specificity, they're also super fast. Like aquaporins could transport up to 3 billion water molecules per second. Also, aside transport, membrane proteins also work as enzymes. They work for signal transmission, cell-cell recognition in the case of glycoproteins, and so on. Another type of lipid that is essential in a cell membrane is cholesterol. Cholesterol is also amphipathic, like phospholipids, but it is mostly hydrophobic also, and so it is found in the hydrophobic tail region. Just its head, the tiny hydrophilic part, interacts with the head of the phospholipids also because it's hydrophilic. So this cholesterol has a couple of functions. First, it helps restrict passage of underage kids, as we call them, by causing the phospholipid tails to pack more closely. But depending on structure, cholesterol could also adapt in various ways, and so it's like a fluidity buffer. And this is important because it helps to maintain the integrity of the cell membrane. If the cell membrane is too fluid, then the phospholipids and proteins will be moving too rapidly and it might just destroy the structure basically. And if it's too rigid, then it means they can't move at all. So either of these extreme cases would interfere with the function of the cell membrane, which is essential for life, and this is why we need cholesterol. At high temperatures, because we know that energy is directly proportional to temperature, the membrane has a tendency to be too fluid, and by too fluid we mean that these components that make it up move too fast and too much basically. And so here, cholesterol interferes with the movement of the lipids and co, thereby reducing fluidity of the cell membrane as a whole. At low temperatures, however, because once again, energy is directly proportional to temperature, the tails have a higher tendency to be more closely packed, but the cholesterol once again interferes with this, and so here it is increasing fluidity. Because interaction between the tails, the phospholipid tails, also serve to decrease fluidity. Kinks in the tail, these kinks that are as a result of cis double bonds, also increase fluidity because they prevent close packing. Other components of the cell membrane are proteoglycans. Who could have thought a word would be so hard to say? Okay, we have proteoglycans for cell-cell addition, glycolipids for cell-cell recognition, and also glycoproteins for immunity. Now, glycoproteins are like the Achilles heel of this whole system because it is through them that a virus usually gets into the cell. So that's it for today, guys. Hope you had fun learning about the cell membrane and the components of the cell membrane and their various functions. Please remember to like and subscribe, and see you later, guys. Bye.